thermophils. It's a creature, it's a bacteria, right? Can survive in over 80 degrees Celsius hot spring, which is very curious. The creature can survive in such hot place. And then this biologist simply curious why this animal can survive in that hot spring. And then try to search for the truth. But this guy, Carrie Millis, who is a biochemist, invent a PCR after extracting the DNA polymerase from these thermophils. So he is an inventor-like person, and he is a scientist person. Obviously, the biologist is this inventor is more rich than the biologist, but they have different goals and then purpose, and I think they are both happy after these very good findings. And then also some people mix up these two words, scientist and critics, so which is also different words. Okay, the scientist, again, the main purpose is searching for the truth. And then the most important thing being scientist is what I think is the no human ignorance. So we must respect the nature and then ask the nature. So to ask the nature, we must know our ignorance, how ignorant we are. And then old Greek philosopher Socrates also say that I know nothing. That's very famous word of what Socrates says. So being like ignorance, it's kind of strange, right? So scientist is ignorance is not that most people don't think that, but the scientists themselves must be know their ignorance. That's one of the important parts. But the critics is different. Critics have a lot of knowledge. Read a book or paper or article, a lot of book, but they don't create a new things, but they explain to someone's logic, just summarize many people's opinions and then explain their logic. That's a critics. So the scientists and critics are very different. And then, of course, the critics makes more money because like they consult the companies or with these knowledge. The scientists do not earn a lot of money. But again, the scientist is more, I think it's a very fun profession in human history. So I recommend to be a scientist. Of course, recommend critics or inventor to, earn more money. But another aspect, the scientists have more good part in these professions. And then, so then back to the talk, the how to cultivate curiosity and creativity. So now I talk the difference of science and technology, so inventor and scientist, and also difference between scientists and critics. And then the scientist needs more curiosity and creativity to being scientist rather than being critics or being inventor. So you must know these two ability is important to being scientist, and then probably you want to know how to cultivate these two ability. And like a business textbook say that the PDCS cycles is more effective. So probably guys, this PDCS cycle. P is as in plan, D as in do, C as in check, and A as in act. These cycle, so first plan and then try and check the what is wrong and what is good, and then just make change, and then another make plans. So this cycle is cursory works in science, but not always works. It works in like business math because it's just create an improvement, right? More after the over cycles, you get nice the technology or invention. But this thought is not always working science or scientists. So what I really important to be a scientist is these two parts. So logic and illogic. So logic and illogic. So most people or student knows that logical thinking or logical explanation is very important to study science. And probably you guys are taught by your teachers say, what's your logic or what's your writing is not logic. So the logical part is very important, but I want to say in this lecture is illogical part is also important to to cultivate your creativity and curiosity. So again, these two words are totally different, actually opposite sides, right? Illogical and logical. The logical in science is required for uh, planning the experiment and also explain the data to other peoples. So you must be logic, right? So this logical part is important, and you know that, right? But illogical part is also important because illogical part create your curiosity and your creativity and your instinct. And these logical and illogical uh, thinking thoughts are interplays each other in your brain and then make a new breakthrough. So eureka. So that's the most fun part of this what scientists doing. And then so most people don't know how illogical is important. But I want to say this illogical part is very important. So then again, you kind of understand the illogical part is important, but how to be illogic? I think it's really difficult being more logic, right? Being illogic is like, it's very ambiguous, right? So then I will show you how to be illogic in your brain. So this is today's take-home message to you guys. And then this one, to break the norm. So break the normal is the most that is the way to be illogic, right? It's kind of difficult. Break the norm is make a new breakthrough. To make a breakthrough, you need to be illogic, right? It is logic, right? And then but it's kind of hard to break the norm in a short time. But the most easiest way to break the norm is question your belief. So that's the I think motivation of scientists. And then question after question your belief, you you try to make a plan to assess whether your belief is correct or wrong. And if it's wrong, you can break the norm and then create a new norm. Once you create a new norm, the norm lasts suddenly and then lasts for a long time. So that's the breakthrough. So anytime you talk with your friends or teacher, you just question your belief or what teachers believe as well. And then discussion is more valuable and fruitful. So then this thing is most important is that, and also that this word is most I want to say in this lecture. So it's half hours is passed. So it's the next topic is just example or expression of curiosity and creativity. So now you kind of understand how to cultivate curiosity and creativity, but no example is not good. So let me show some example using my research topic and then how to express my curiosity and my creativity into my project. Okay. So again, I'm a eel researcher, eel fish researcher, fish biologist, and then but I'm not a typical fish researcher. And then as Professor Chimisan introduced, I have many backgrounds. I was in Ocean Research Institute of University of Tokyo, and then at that time I studied fish biology, and then I moved to United States to study ESL, embryonic stem cell, and then I studied cell biology and regenerative medicine. And then back in Japan and met Achimi and Reza-san, and then we study histology. And then I own my laboratory in 2016. And then I asked by myself, and then what I have to do in this university. So I can pick any research field, but I want to do the world only one laboratory. And then I relaunched a new eel biology since 2016. And then it's named neo-anguillology. Neo means new. Anguilla is a Latin name of eel. And then LOGY is the study, the field. So I established a neo-anguillology. So if you want to study neo-anguillology, this is the only lab in the world, and then you must come to study neo-anguillology. So then, before I explain the neo-anguillology, I need to explain what's the typical classic eel biology that you guys mostly imagine. Most people think if I do fish research or eel research, most people imagine I'm studying like field investigations, like I'm an ecologist and then do a field investigations. Or some people think it's a artificial cultivation, aquaculture parts, because eel cannot be fully artificially cultivated from egg to spawning. So most eel biologists study this artificial cultivation of eel. And then eel is also endangered species. This is the quote rate of Japanese eel. Then it's very small letters. It's 1957 and then 1981. After 1981, the number of eel is really decreased, and now it's a list of endangered species. So some of eel researchers study this management of these fishery resources. And these like ecology, aquaculture, and management are the typical eel biology. And then most people easily imagine how eel biologists do. But I'm different because this is a new eel biology. So my goal in my laboratory is extreme translational research using eel. This thought is kind of crazy thought. Nobody thinks that eel can help human disease, right? So I question my belief and then I just break the norm. So eel can help human disease. And then this is my this the words, the ill to bedside. This is my final goal, but nobody do. I'm not sure this works or not, but let me explain this may works in using my research. Okay. So then how this ill to bedside research works. So most people don't believe how ill can help human disease, right? And most smart researcher using these good model animal, like if you use a fish, some people use the zebrafish or medaka. These are genetically very organized species. And also most biomedical students or biomedical researchers use mouse or rats, right? And nobody use eels. And because why these people use this animal model? Because they are really smart, because it has a short life cycles and small and cheap and easy to manipulate. So when the life cycle is short, you can see the development, right? Of the embryo to adult. And then if the size is small, you can keep this animal in a small laboratory. And if it's cheap, you can save the budget. And also if it's easy to handle, so like genetical manipulations, you can investigate the effect of the gene or influence of the gene when the gene is deleted or overexpressed. And most important thing is this animal has a commonality. Commonality means what you see in like medaka or mouse can be applied to the same thing in a human. So because it's the physiology. So that is commonality. So that's the very good model animals, right? The short, small, cheap, easy, and commonality. Now everybody can easily understand these animals are useful, and then eels are not. And then, and most people know that eel, the fish, is a non-model animal, and most people don't use this because this has a long life cycle. The eel's life cycle is still mysterious, and we don't know how long they live. And it says more than 10 years, 10 to 15 years life cycle. So if I want to see in natural development of eel, I must wait 15 years or 16 years, more than 15, 16 years. So it's not good to study a development of eels. And then size is middle. It's not a big, but it's middle. And then it's very expensive. It's about 10 US dollar per eels. Actually, this medaka is like 50 cents or so. So it's very expensive. And also it is very difficult to handle, especially genetical approach. To manipulate the gene, you need an egg of eels. But again, the eel is not fully cultivated artificially, so we cannot obtain the egg from the eel. So it is very difficult to manipulate gene manipulation. And then eel is very peculiar. Peculiar means it's very special. And then what you see in an eel cannot always be applied in human or medaka other fish. So what we found in eels is just discovery in the eel. It cannot be applied other animal because it's very peculiar fish. So it's not good parts, using eel as a model animal, but I focusing on this peculiar character of eel, peculiar. And then what I'm doing in my laboratory is extract a universality from peculiarity. It's very difficult, like logic, right? Most people understand extract universality from commonality, that makes sense. But extract universality from peculiarity doesn't make sense. But I do this approach. So find the peculiarity and then extract universality from their peculiarity. So here's some biggest breakthrough in biology. I just showed 3 examples. So one is discovery is acetylcholine receptor. So after that, the pharmaceutical company develop a lot of drugs using these structures. And also this one is discovery of action potential of neurons. And this one is discovery of green fluorescent protein that is used in many applications in biology, chemistry, and also like biomedicine and optics a lot. And then these are like a Nobel Prize. This one is Nobel Prize, and this one is probably Nobel Prize or just Nature science papers. So biggest breakthrough. And these findings contribute to human disease. And then, but this discovery is not from the human research. These discoveries are from these unique animals. So the first discovery of acetylcholine receptor is from marble electric ray. And first discovery of action potential of neuron is from squid. And then first discovery of green fluorescent proteins from crystal jellyfish. So not a human research, these unique animals. Why? Again, these animals are very peculiar. Marble ray have electric organs, and squid have visible giant neurons, axon. And then crystal jellyfish has fluorescence. It's a very peculiar, and none of character is not found in human, right? But my logic is convinced after I see this discovery. So it can help human disease. Again, so then to extract the universality, I need to know the peculiarity of Japanese eel. So this is the life cycle of Japanese eels, and then eel is spawned in northern Mariana Island, is a deep sea, more than 800-meter depth, and they spawn and egg. And then after hatching, they first be a juvenile fish, Leptocephalus. And the Leptocephalus is very flat body shape, and then it's float in this crucial column and leads to landing in Japan Island or Korea or Taiwan, China. And then after landing, they transform metamorphosis and then become like very tiny, like a fish. And then in the liver, they grow in the liver and then named yellow eel. So the body color changed white to yellow. And then after a growth period is done, they change their body color to silver to migrate to the sea for spawning. So the peculiarity of eel is the migrations, large migrations, from Philippines to Japan. It's 2,500 kilometer distance. So these guys migrate very long journey. And then they can survive in seawater and they can in land, which is liver. So they can live in 2 different extreme salinity environment, which is peculiar. So if you keep in the seawater fish in the freshwater, most fish died within 30 minutes. And if you put in like a catfish in freshwater into seawater, the fish dies in 30 minutes, vice versa. But eel can survive both salinity. So they have a very tolerate to salinity. And also these guys are metamorphosis, so change their body. That's also peculiar. But I'm going to show some example today is a tolerance of starvation. So it's very ill is really tolerate to starvation. Starvation means not hungry, starved. So let me talk about the starvations. So I hope this YouTube movie works. So this is a cultured eel, and then this mud is a bait of eel, and then eel have very extreme hunger when they in this fish pond, so they eat a lot and in a river because it's a growth phase. But after that, if you add a cold stress to this eel, they stop eating, a lost appetite. So they don't want to eat for a long time. And this movie is my laboratory fish named Sakura. He lost appetite for 1 year and 7 months. So they survive without food, bait for more than 1 year, which is very peculiar, right? So after this stress, they stop eating. Even if I give some pellets or some living animals like insects, they don't interest at all. They don't eat at all. And then they can survive one average wise is 1 year, but this guys is survive 1 year and 7 months. And then after that, I just weigh this Sakura eel, and then I found this eel lost 100-gram muscle. So when I bought this eel, it was 200 grams, but after 1 year and 7 months, the weight is 100 grams. And then I opened their stomach, and then I found these tissues, and then liver size are very decent, but the muscle size mass is very small. So means most weight loss is attributed to the loss of muscle. And then muscle is eating part. So eel is eatable. And then I calculate the energy of 100 grams of muscle, which was a 255 kilocalorie, which means this eel can consume only 255 kilocalorie energy during starvation. And then how this energy is just one bowl of rice is equivalent energy. So they can survive just one bowl of rice for 1 year and 7 months, which is really a peculiar and then super metabolic. And then I'm studying this appetite. So why the eel stop appetite and why this eel have a special metabolism mechanism. So I'm studying this. So after cold stress, probably stress get in a brain, and then after that, a brain and digestive tract interaction, brain muscle interactions, and brain liver interactions. So metabolite change. So they stop appetite and change the metabolic rate. So extraordinary appetite inhibition and lower metabolism are caused by these tissue interactions. And then I'm studying these interactions. So how the eel stop appetite and how eel metabolites works during starvation. I'm not going to show the detail, but after these findings, again, this I'm going to apply to a human disease, right? Stress, appetite loss, lower metabolism in eel. It's same thing happening in humans, right? Eating disorder after strong stress or getting more fat after strong stress, or we cannot control appetite after strong stress. So if I understand this extraordinary appetite loss or lower metabolism in eel. This finding can help these disorders. So this is one of the breakthrough things. So break the norm. So if you want to, most people think if you want to help human eating disorder, you just go to see the patient. But I'm not. I'm just look for these eels and then just think the another resource for this study. And then back to the peculiarities of eels. And then next, I'm going to talk some serum toxins. So eel is toxic. You don't know. If you eat uncooked eel, probably Japanese eel uncooked fish like sashimi, but probably you guys don't eat uncooked fish or eels. But if you eat uncooked eel, you get diarrhea, vomiting, skin rash, and sometimes you get anaphylaxis. And then because eel serum contains toxin, and then they have been long known that eel have toxin in their blood, but people don't know what's the toxin in the blood. It's not been found yet. My laboratory, I found the eel toxin gene, and then I purified it. And then I inject the purified eel serum toxin into the mouse here. It's don't move a lot. This is control mouse. It's move a lot, and I inject the heat denatured eel serum toxin. So this is a protein toxin. So once you denature it by heat, it's detoxinized. So these guys can move a lot, but when I inject the eel serum toxin, they get sick. So toxin itself is bad for humans or animals, but we call toxin because we don't know the mechanism of how toxic these molecules are. But once you know the pathological process of this eel serum toxin do, you can create a new drug, right? So this is another break the norm part in my example of my study. So ideas are infinite with your curiosity and creativity. This is the words I want to say in this lecture. So you have a very infinite curiosity and creativity, but if you don't know how to use this, and then once you can get this ability, your idea are infinite. I have more other projects. I mean, its time is almost close, but I'm not going to show a detail, but especially so I used to work on a regenerative medicine. So then ill and metamorphosis do a metamorphosis. So if I understand, I can contribute to regenerative medicine from research in eel tissue remodeling. And also, I'm an endocrinologist. I do a metabolism, hormonal regulation of metabolism, and also reproduction of eels. So this research may help conserve species and increase fishery resources. Okay. So then the question you brief is not very difficult, rather fun. And then this, again, you can find such this peculiarity character. You can make a big breakthrough after break the norm. Thank you for your attention. My talk is done. I can take any questions if you have.
Okay, thank you, Sensei. Everyone, can give an applause using the reaction? And you can also raise your hand if you have questions for Dr. Sukada. Oh, we have a question from here. Okay. First, let's take a question from Anissa Delvira. Anissa? Anissa, you can turn your camera on. It's a really an amazing talk, Dr. Sukada. I really enjoyed it. Our target audience is the graduate students, but I get a valuable insight this morning. Okay. I also see that Dr. Laila Isrona raising hands. Please, Dr. Ona, yeah, if you have questions.
Dr. Chime, I'm sorry, I'm not raise hand, but
Okay.
I give applause for.
Oh, you were clapping. Okay,
yeah.
Thank you, Dr. Ona.
Thank you very much for the very amazing presentation and material for us. Yeah. Arigato.
All right. Thanks.
Okay.
Reza-san raised hands.
Yes, Reza. Dr. Reza, please. Dr. Reza, you're still mute. You're still mute.
Okay.
Okay.
Sumimasen. So, this is very interesting because you talk about breaking the rules or to break the norms since maybe accidentally I get used to break so many norms and rules, but I don't know it's related to the science or not. But yeah, it is very interesting because your lecture is just broaden my knowledge. That is sometimes breaking the rules or breaking the norm is very important to generate curiosity or creativity. So I don't know if it's related or not because yeah, it happens. Because nowadays the students tend to follow the trends. So they would like just to what is the trends for today's research? They just follow it. They never think about something new, something is not approachable, or something maybe not thinkable before. So yeah, but for example, I also have experience on that occasion with my research because before when I do research in mosquito, after I moved to the entomology department, so I also maybe do your methods without realizing it. But now I know because you said breaking the norms generate curiosity and generate creativity. Before I just think maybe by accident I just think about something stupid, just how to use copper to kill mosquito larvae. But yeah, accidentally maybe I do what you just taught today. So, but I just cannot how
Question,
to
Dr. Reza.
yeah how to
Please
make
shorten it.
how to make the student realize because how to make the student realize by breaking the rules sometimes can be better rather than just following the rules because today I just see the student is just tend to follow all the rules, all the norms that in research, all the trends.
Okay, Dr. Reza. So we get your questions, how to inspire students to break the norm. Please, Dr. Sukada.
Okay, thank you, Reza-san. So that's really difficult part. It's the student must be diligent. It's good, but breaking the norm is kind of problematic. But both thinking it requires again a PCDS cycle to improve your thoughts. The one of the important parts, and also like free thinking that comes from the curiosity. So to make a atmosphere to discuss in your lab, that's in both sides. That's important. So not only are diligent parts, also very fun parts, illogical parts and logical parts must be get together in one laboratory. That helps the student more creative and then have more curiosity.
Okay, so the conclusion is I think we can conclude that sometimes it's good to be illogical rather than
Right.
logical.
Right.
Thank you. Thank you.
I thank you.
Okay. Okay, thank you, Dr. Reza, Dr. Skada. Is there any other questions from the students? I'm really expecting students really get some insight or maybe some questions. Dr. Skada, I think it's so interesting that how you give examples, you have applied the logical and illogical parts to break the norms, and you have conduct this such way of thinking in your own research. My questions is this is not seems like you so many things against the odd when you use this peculiarity of EL, right? And nobody thinks the way you do, and you establish the only EL laboratory in the world. That's amazing. But I'm sure it's not an easy task on your part. It seems like you're against the odd. And can you share some of the challenges when you break the norm?
So some signal was noisy. I couldn't
Oh,
hear
yeah.
the last parts of your questions, the most important parts.
Okay. What are the I'm sure that you have challenges when you break the norm. Maybe you can share some of the challenges and how you overcome them. So now you have this established and unique laboratory.
そうね。So why I get this thing is kind of very difficult because I'm actually first reason is my laboratory is very small. So in Japan, there are more huge laboratory in Tokyo University or Kyoto or Jichi, also big. But my laboratory is very small. So if I do the same thing, so there are many competitors, right? So I must be more creative and then to fight against these big laboratory. So I must be a peculiar and must break the norm. This is the basic thing. Just my atmosphere do this.
Okay, and if you now that you have this insight as a professor about cultivating curiosity, creativity, and all these experiences, if you can go back in time when you were a graduate students, what advice would you give to your younger self?
So question what your teacher says. Yeah, I was
Questions your teacher.
Yeah.
Okay.
Yeah.
I think I already do that so long time ago.
Yeah. Okay. Okay, students, if you're still listening here, don't forget to question your teachers, including me. Okay. Is there any? I don't think these students they have responded in pre-test, but yeah, maybe there's it's still a new thing for us, Sensei, to have a guest lecture like this. So, but I hope you have inspired them this morning by your talk, as you have inspired me, and we can just wrap up this lecture. Before that, I'm going to share a link for post-test for the audience about 5 minutes. And if you can hang on with us, Sensei, for 5 minutes, then we will close the lecture and we'll take picture together.
Sure.
Okay, thank you so much, Sensei. Okay, I'm going to share the post-test for the audience. Please fill this post-test if you want to get the certificate for this guest lecture. Okay.
Jimmy-san, please also share the link in the clickable link because I use mine.
Oh, yes, of course. We have a a group for the audience. We'll share the link also in the group. Okay, now I think it's time for the closing. Thank you so much, Sensei. There's so many take-home message, but please, students, remember that to cultivate curiosity and creativity, you have to have the courage to question your beliefs and to use the illogical part along with the logical part in your learning and experiment, and just don't be afraid to break the norm and be inspired by previous scientists that has paved the way. And remember that science and questioning beliefs and breaking the norms can also be fun. That's, I think, the message from Sukada Sensei. And one more thing, Sensei. I still remember more than decades ago, you told me that one day, I mean, it's a dream of every researcher, right? That you want to get a Nobel Prize, right? It's a dream. It's a ultimate goal. I still remember that. And I can see that you're making your way with this unique and peculiarity mindset. Thank you so much,
Thank you.
Sensei.
Thank you, Jimmy-san, for inviting
Thank
us.
you, everyone. Let's
Bye.
take picture. Everyone, if you can turn on your camera, please turn on your camera. And let's take picture. The first page. Okay. 1, 2. Give your best smile. Okay. Tami, would you help me with the taking of picture? Okay. 1, 2, 3. And the second page. 1, 2, 3. Last page. 1, 2, 3. Okay. Okay, thank you, everyone. Let's give the last applause before we send off Dr. Sukada. Thank you so much, Sensei.
Sensei,
Thank you.
arigato. Can I ask your
Arigato.
contact from Jimmy-san? So maybe next year I will visit Japan. I will very, very love to see you and see you.
Okay, see you.
Okay, no worries. Thank you, everyone. Bye-bye, Sensei.
Bye-bye. Good
Everyone,
day.
have a good day.